Hello, my name is Justin Nam. I'm a rising senior at Rutgers University and IMSD student. Today I'll be going over the polio virus genome. We will be discussing its structure, the role of its proteins, and replication. The polio genome is a positive single-stranded RNA. It includes a 5' and 3' untranslated region with a long open reading frame in between. Polio has many structures in its untranslated region that will play an important role for genome replication. The iris is an internal ribosome entry site. It is necessary for translational initiation of the open reading frame. The viral protein genome link, or the VPG, is a protein that covalently attaches to the 5' and acts as a primer for gene replication. Polio also contains 3 types of cis-acting replication elements, or CRE. They are important for positive stranded RNA synthesis. This includes the clover leaf on the 5' end, the oral on the open reading frame, and the pseudoknot on the 3' end. The open reading frame will be translated into a single polyprotein, which will subdivide into smaller polypeptides, which will help and aid in replication. P1 includes the capsid proteins, P2 will help in host cell takeover, and P3 will help in genome replication. The polyprotein splits into many proteins with different roles. Some proteins of note include the 3D protein, which is an RNA-dependent RNA polymerase that aids in genome replication, 3CD and 3C, which cleaves most of the polyprotein, and 2A, which has a major role for degrading many host cell processes. For example, protease 2A degrades the host cell translational factor, thus leaving translation ineffective. Protease 2A also blocks mRNA from leaving nucleus by cleaving its nucleoporins. The first step in genome replication is transcription of the original positive-stranded RNA, which will produce many negative-stranded RNAs. The negative strand will serve to produce more positive strands for progeny down the line. Looking at Figure A, negative strand RNA initiation begins when 3CD binds to the 5' cloverleaf in the 3' untranslated region of the positive-stranded RNA. They come together to cleave 3AB and 3CD at the site of replication, releasing both a tyrosine hydroxyl VPG and 3D RNA polymerase for elongation of the negative-stranded RNA. The next 3 steps goes through the formation of the VPG-UP-UOH, which will prime the positive-stranded RNA initiation, which will persist until there is enough progeny for lysis. Thank you for listening to my talk.